So, good afternoon, everyone. Can you please confirm if you are able to hear me and my video is accessible to you?
Good afternoon, sir. Yes, you are audible and visible.
Thank
to
you.
me, at least.
Okay. So I think few new students here today. So I welcome you all to the session. And I'm assuming that you probably have already seen my previous lectures, last two lectures after orientation class, two lectures, one on the DNA basics on unit 1.7. And we also talked about cell structure, cell division. So last class we focused on talking about cell division. Okay. So people who are comfortable, you can please on your video lecture so we can actually feel a bit more connected. That is something we are trying to do in these lectures. Okay.
Is there a possibility to enable background blur for the video sessions? That is the feature that you as a host need to enable.
So what is it again? I'm not aware about it. You're saying enabling background blur.
Yeah, it allows to blur the backgrounds or put some kind of effects.
Okay. Do you know how I can do that? You edit window. Any idea like how this needs to be done?
Just checking, sir.
Okay, fine. I've really not played very much with this, but Zoom I've been using since the beginning of lockdown, but I haven't really worked on features like this. Okay, anyone have any questions to begin with as we are figuring it out and from the previous classes? Especially students who newly joined.
Sir, I have a question. So this is regarding meiosis actually. So we said that for the mitosis we have 46 and then they divide into 46 and that happens in somatic cells. But for meiosis we start with 46, but then again it duplicates into 46 and then it divides into 4, 23, 23, 23, 23. But my doubt is like, let's say there's a male and he has, let's say a gamete and he has 23, right? From the beginning. Then and for the meiosis, why did we start with 46? Shouldn't we have started with 23?
Yes, I see where you're missing it. Okay. So your question is, if you're talking about production of gametes, where gametes should contain 23 chromosomes, that is one set of chromosome, why should we start with 46, not 23? Because what we are talking about is the process behind production of those gametes that you are mentioning. The gametes, the process of meiosis is what results in these gametes. So in the first place, we don't have them, correct? So these have to be made from the existing germ cells. So the sperms and egg cells, for example, they are not made until we attain the puberty. So initial cells required for it are made when we are born, but they don't complete the division. So what happens, they're actually kind of stuck in the cell division process. Only when we attain the puberty, that is when we start seeing that gametes are being formed. So that is when process of meiosis kicks in. Okay, so the initial product of this process is the gamete. So unless meiosis happens, no gamete is made. So if you're assuming that gametes are made from gametes, then your theory is correct. So then we are beginning with the gamete, but we're not beginning with the gamete. We are beginning with the cell which has a capability to produce gametes. Like, in case of males, for example, these are formed in testes. So there need to be a cell division that is taking place in the cells present in the testes, which also are diploid. So only from these cells, in the process of meiosis, gametes are formed. So the result of meiosis is this gamete. So we are not replicating the gametes here. We are creating gametes from the other existing cells in the germline. Is that making sense? Or
Yeah, blurred? It's getting a little clear. One more thing. So the germ cells and the somatic cells, so there's no difference in the number of chromosomes.
To begin with, so germ cells not only contain gametes, germ cells will have other tissues as well in them, but which undergo meiosis to give rise to gametes, like testes or ovaries in males and females. They show the meiosis to produce gametes. So only during gamete production they use meiosis. Otherwise, they also replicate like other cells. They also have mitotic divisions happening in them. But meiosis, what actually results in the gametes when it happens in the germ cells. Okay,
And these germ cells are only found in the reproductive part.
exactly. They are only found in the reproductive parts, especially in the ovaries in females and in the testes in males. Correct.
So, right, if someone grows up, still there would be 46 chromosomes or 23 chromosomes since he has grown up now. Or, I mean,
Only in the gametes. Only the gametes. Only sperms and eggs will carry the 23. Rest all the cells in the body carry the 46. So are you seeing the point here? So whole body actually has 46 chromosomes. Only when gametes are made, even in the germ cells, only then they end up having 23, meaning only one set but not two sets. That's the only process that undergoes meiosis in whole body.
so there must be germ cells which have 46
Yeah.
and okay. So
Germ
together
cells
they
also,
exist.
exactly, they do exist. They exist with 46, they multiply with 46, but only in this one particular process called gamete production, they would follow meiosis, resulting in something that has 23. So that when they really pair, they give rise to again 46 chromosome containing diploids. So this is how we are able to maintain the chromosome number constantly. But only situation where we have to reduce it is the gametes. Until that step, even till the formation of gametes, it is actually following mitosis. So mitosis is connected, but just to make it clear for the student, we have to say that germ cells actually undergo this meiosis, then resulting in this. But even germ cells are replicating. When they're replicating, they're following mitosis. But when they're giving rise to gametic cells like this, they're following meiosis.
Okay.
Kind of making sense?
Yeah.
Look at a
I
real
got.
picture, but this is bit complex, but you have to have basic understanding. That is why I'm spending time on this, because although they don't come in the exam, but this idea is very, very important. When we are talking about diagnosing problems in these type of conditions, we need to know where and how we are diagnosing the problems. So this to me, because even some rankers I've seen that they say that I left your unit 1.C because it is not going to come in the exam, because they're failing to understand the connection between what are all they're studying and the link to unit 1.7. Because you have these concepts clear to you, you will understand the applications better. And that is why we are spending time to give you that picture. And especially if many of you are not from biology background, I want you to get a feel for this. Okay. And even if you're unclear about it, now your picture is slowly becoming clear. When you read more, when you start referring to the articles I'm giving. And if you tell me where exactly you're kind of stuck, for these classes, I'm also bringing videos. So with animations, where you can see animation and get a better picture about it. Because even if I speak to you for half an hour, sometimes it may not help because you're not getting the right picture, but you're getting the details. Then if you put an animation before you and put this information on it, it becomes easier. So today I'm going to try videos along with my teaching.
Yeah.
Okay. Any other questions, guys, before we continue with the next topic today? Yeah, all good? So, okay. So please feel free to stop me wherever you're stuck, because this is the area where I think we are getting used to each other. And second, we are trying to figure out something that may sound very complex to you. Okay, but you are clear with this information. Most of the topics that we are going to study next is a matter of adding information, adding principles, but we are forming the foundation for all of it here. Most of the physical anthropology topics require this class, this understanding. Okay, this last 2-3 classes of unit 1.7. Okay, but don't worry, you may even require a little bit time to get used to it. Recognizing is different, using this in your answers is different. So that may take a bit time. Okay, right. Okay. So Vishal, did you figure out what needs to be done for the background blur?
Not really, sir. It's very complex here. Though it's easier to do on Google Meets. Seem to be you need to have a very constant background color. So for you it might be easier, or I think even nickel it might be easier.
There needs to be some provision in the type of camera you are using. Some green feature is there.
Green screen is the background that you have. you need to have a
It is blurred.
constant background.
Uh-huh.
Then
But
you can blur it.
if I do that, will that be blurred for everyone, all the participants?
No, sir.
On my side, right? It will only blur my background.
Yes, yes.
All right.
Yeah. So you can see what I've done. So this is my video right now. If I apply some filter, now it kind of changes. But there is a lot of noise. So many things in the background.
Correct. Okay, let me see. Yeah. So are you having a disturbance with my background? Do you see too much of light? Is it
no, sir.
troubling you? No? Good. Great. Okay. Should we get started then? Anyone? Sunil, do you have anything? Sunil or Nikhil? Are you good?
It's okay. I got mine down. Okay.
Perfect. Okay, guys. So yes, we finished talking about cell division. As I was telling you, don't try to by heart anything from this. This unit to make you familiarize with the concepts and a basic understanding about what we are going to learn next. So even if it sounds complex, understand what it is. Don't try to by heart it. Be familiarized with what are chromosomes, what is DNA, what is genes, how our DNA is actually created. When you really read what is genome sequencing studies, or when you read about how we are diagnosing things, this information will give you a better connection. Okay, you will appreciate this information once we get into the applied part. Okay, right, guys. So today's class, I think we are left with two topics. One, we are going to begin with DNA replication part. Second, we are going to talk about protein synthesis. A brief picture about what happens, the process behind it, and where does it happen, and what is the significance of this process. So in a way, today's topic, DNA replication, ties back into the concept of DNA replication. If you all remember the cell division, cell cycle class, so we discussed that there is a preparatory phase and there is actual cell division phase, correct? In the preparatory phase, what normally happens is cell is preparing towards the division. So the main preparation that we talked about happens in what is called S phase, or DNA synthesis phase. This is where DNA gets doubled. Correct. So the doubled DNA gets separated between cells. When new cells are formed, each of them receive 46 and 46 chromosomes. Correct. So here we are zooming on to that synthetic phase. What exactly happens during that synthetic phase, specifically in connection with doubling of DNA? This is what is called DNA replication. Replica means an identical copy of something. So we are creating a replica or identical copy of something in a process called DNA replication. Excuse me. Right, this is what we are going to talk about. So let's see how DNA gets replicated, what steps it has to follow, and some details about this process. This is where we are. Okay, this is how it is connected to the last topic, that is cell cycle and cell division. Okay, first of all, let me actually pull up here the PowerPoint slide that I wanted to show, and then in between I'm going to show you a video. So video will give you a little bit of idea about it, but first I'll provide you a brief summary on what DNA replication. If you don't develop a complete image on that, we'll watch the video and then we'll have a discussion on this, so that we'll go back and forth between the slide and the video. Thereby, in next few minutes, we'll figure out what DNA replication is. Okay, right. And wherever you are stuck, your imagination is kind of stopped or paused there, do let me know. I'll try to give you the right picture about it. Okay, right. Okay. Right. So now we already know what is replication, right? DNA replication. It is making additional copy of DNA before cell division happen. That is what is replication, right? So it should be easy and straightforward if you really followed my last class. So where is it happening? It is happening exactly in the DNA synthesis phase, just before the cell division that is going to take place in preparatory phase called interphase. Correct. So DNA synthesis or replication is making copy of DNA, and which takes place during S phase of interphase. S phase of interphase. Okay, if I have to take you back to that image, So this is what we talked about in cell division, where we had a G0 here, G1, S, G2. Then mitosis or meiosis has taken place here, right? So this S phase that we are focusing on, the phase where DNA got doubled. Okay, the process here is called DNA replication because DNA is making an identical copy of it. Here exactly the copy we have in the cells, a new copy is made from that. Okay, right. So how does it happen and what is needed? What type of components are required for DNA replication to take place? So DNA is a double-stranded structure, right? So if you remember from chromosome, if you pull the DNA, so DNA is a double-stranded structure. It has two strands, and we were talking about in the DNA's basic structure. So each strand has those nucleotides because it's a polymer of nucleotides. They are made up of sugars, phosphates, and bases. But we recognize each nucleotide based on the base, whether it is A, T, G, or C. Correct. Okay, so first part when we're talking about it, try to understand because you will only know what to write when you understand the information. This is one thing I always had a difficulty to convince students that, as soon as even me, if I'm sitting in a class, as soon as I hear something, I like to write because we may feel like we are going to miss something, right? But I think the most important thing here nowadays, especially because we have enough data in our hands, we have information resources. So it's a matter of understanding is what is most important. So focus on understanding it. Once you piece together the information, now you know how to write it. That is the most important. Okay. If at all you want, write the key terms in between. The terms I'm using, if at all you are worried that I'm not going to repeat. Okay, but anyway, you also have access to the video, right? You can repeat it and see if you missed anything. So even that can be written here. So focus on the concept here, then focusing on writing for the first part. So DNA replication means making copy of it, and DNA is a double-stranded structure, which each strand is made up of nucleotides. So what happens here? So this DNA, in order to undergo the copying, what happens? Each strand acts like a template, acts like a guide. Because what we are trying to copy here, we are trying to copy the information, we are trying to copy the information on the DNA because DNA brings instructions in the form of nucleotides. So here our goal is to just copy that information, just copy the wording, just copy the letters in that order because there are only 4 letters ATGs, but the order is going to make the sentence. Whether you put ATG together or ACG together, GCG together is going to define the sentence, is going to define the meaning of that instruction, correct? So what we are trying to copy here, exact order of those 4 letters as it is present on your DNA in the cell, which you have received from your parents, correct? So the goal is to copy both the strands. To maintain both the DNA strands, you want to copy both the strands. But if you want to copy strands, imagine it's like in a Xerox that you want to make a Xerox of it, but it's a 2 sheet, okay? Attach it to each other, pin to each other. What do? You actually unpin it and take each of them and make a Xerox of it, right? Make a copy of it. Similarly, this double helical structure has to be first opened up so that we get 2 strands and copy each of these strands to prepare the new strand. That is what happens here. That is what you're seeing in the structure. So this DNA is actually unwound. Okay, you took it and you unwound it and kind of ripped apart. Okay, or separated. There are specific words that are used for this. In the general scientific term for this is denaturation. Renaturation is coming together. Denaturation. I'll type it here. This process of DNA strand separating is called denaturation. Denaturation means separation of 2 strands. Separation of 2 strands. Okay. The first part here is for the DNA, double helical DNA strand has to unwind. But how does it get unwound? We need an enzyme to do it. Okay. So this process is facilitated by multiple enzymes, but I'll focus on only few enzymes. In the video, they talk about more enzymes because they make it a bit more complex. But see that we need proteins because enzymes are nothing but proteins. Except I think one example that we're going to talk in the next topic. The rest of the enzymes are made up of proteins. Okay. So these proteins acting like enzymes. Enzymes are biocatalysts. You probably studied about catalysts, right? They speed up the reaction. But here we are talking about those speed up the biological reactions. They are called enzymes. Okay. So for example, here we have what is called helicase. Helicase. Helicase an enzyme. What does it do? Helicase enzyme actually helps in unwinding process. So it's like, it is kind of something that is actually going into the center of the DNA, thereby opening it, thereby separating. It's like, something swimming into it and moving it apart, moving the DNA strands apart. Such an enzyme is called helicase because DNA is a helix. Something that removes the helix is helicase. So this is the first enzyme which separates the DNA strands, which opens up and separates the DNA strands. So that is how DNA replication process begins, with separation of DNA strands. And this is nicely shown in the video that you are going to watch next. Okay, that. Now we have access to both the pages. Imagine both of the DNA strands are different pages. Only when you actually open them like that, now we have access to read both of them, both the DNA strands. Okay, right. Now we can begin the process of synthesizing new DNA. We can begin the process of synthesizing new DNA. Okay, for this, the key enzyme responsible. Now I'm introducing second enzyme for it. What is the second enzyme? DNA polymerase is a second enzyme. First enzyme is helicase. Second enzyme is DNA polymerase. Not too complex. Helicase is something that unwinds the helix. DNA polymerase, something that makes the polymer of DNA. Ase is a normal suffix. Many of the enzymes, not all of the enzymes, like protease, nuclease, okay, oxidase. These enzymes have ase at the end. Ase is a common suffix to several enzymes. So here polymerase, what does it do? It is creating the polymer of DNA. That is why it's called DNA polymerase. In the next topic, we also learn about RNA polymerase. The enzyme that synthesizes RNA is called RNA polymerase. The enzyme that synthesizes DNA is called DNA polymerase. DNA polymerase. Okay, right. So this is a key enzyme. This is the most important enzyme for DNA replication process. You know what? Just to put a little bit interest in your head, to understand how does RT-PCR work. You all heard about RT-PCR? You heard about RT-PCR these days? No? No?
Noble.
Yes, sir.
Related to what? RT-PCR, we have been hearing almost every day. You can pick it up in the
COVID.
newspapers.
COVID
test.
testing.
COVID test. This is the gold standard test for COVID. And the whole COVID test, RT-PCR test depends on this process. Because we understood this process very well, we are able to design something like the RT-PCR test for COVID. So this is a principle behind how RT-PCR test works. So even application part, if you're talking about DNA technology in medicine and the usage of DNA technology in dealing with COVID-19, if such questions appear in the exam, this is the understanding that you need for it. Just to give you an additional reason to understand this. Okay, let's see. So here, that is what we are seeing, that we have enzyme called DNA polymerase, which is actually synthesizing the DNA process, DNA. Okay, how does it do?
Sir,
yes.
Sir, only RT-PCR is based on this. Like other antigen and Trna, these are not based on this.
No, they are not based. Trna is also a type of RT-PCR and an automated RT-PCR test. Antigen antibodies are not. No. We study them in immunogenetics. We have one topic in Unit 9.1 on immunogenetics. I'm going to cover those aspects as well. So more or less, you will be able to understand the COVID scenario and the technology behind it better after our classes. Okay, even CBNAT test, have you heard about for TB detection? CBNAT or nucleic acid amplification test. We mimic this process that happen inside the cells, DNA replication process.
Yes, sir.
Okay, right. So next. So we're talking about here.
Sir, just to understand, so since you brought up RT-PCR and connection with polymerase, so here polymerase is helping creating new DNA. So in RT-PCR, does it kind of enhance the, say, the available virus DNA to make it detectable, or is that how it works?
Yes, the machine called PCR, polymerase chain reaction, we use there. It's like a Xerox machine of DNA. So what does it do? It makes the copies of the virus. Even though you have very few molecules of the virus in your body, it will amplify the signal. So it's an amplification process. So we can detect it easily, even with very small amounts. That is why RT-PCR is a very, very sensitive test. Okay, you're exactly right. Those connections definitely you have to make. You will understand it because in DNA diagnostics, we have molecular diagnostics, molecular anthropology being asked these days very much in the exams. So PCR is an example under that. That's a core idea in molecular anthropology. Okay, right. So yes, now we opened the strand with the help of helicase. Now we are actually trying to synthesize the DNA. It's like you copying the information. Like you want to see what is written on the board and you write down. Like let's say letters, only we are using 4 letters, assuming that. So we are writing A, T, C, C, G, G. You just copy that, A, T, C, C, G. This is what DNA polymerase does. It looks into this DNA strand, which we call it template strand. These two separated strands are now called or now act as templates. By looking at each of the strand, you will write down the information. Who is writing the information here? DNA polymerase. And how does it write? It doesn't have a pen, so it has to write it by adding nucleotides. So it's a concept of joining nucleotides to match the DNA template. Okay, so where are nucleotides coming from then? So we're talking about, we are actually zoomed into the cell, right? Every cell as we are speaking is doing this. In almost all cells, not all, but majority of the cells inside our body are doing this DNA replication now. But where are they getting the raw material from? The raw material here is the nucleotides, because we are making the polymer of the nucleotides. So you need to get nucleotides from where then? One, our body can synthesize to a certain extent. Second, our food. We eat either plants or animals, right? Mostly we rely on plants and animals. Plants and animals, do they also have DNA in them? They have DNA. What our body does? Have you ever thought that whether we use plant's DNA, animal DNA as such? Like we eat plant material, every plant cell will have DNA. Are we expressing plant DNA inside our body? We don't. What do? We digest it to a level where we can extract this raw material from it. It's like, analogy if I have to give you, you want to build a wall. What do? You go to the next home, break their wall, bring the bricks and build your wall. So it's like, getting raw material from other sources. Here, our neighbors are plants and animals. We eat them, we get their cells, we break down their DNA into individual raw material particles like nucleotides, and in the process of DNA replication, these enzyme DNA polymerase will take the existing nucleotides in the environment in the cell and just add them. But for that, it cannot randomly add anything, right? We have to define what to add. You have to write what is being told, what is being written on the board, right? So similarly, whatever DNA we have, we make the copy of it. You are not rewriting the information, you're just copying the information you already have. So the same DNA information that you have from birth will continue till our death. Except once in a while we make mistakes in writing. Instead of writing A, sometimes you may write T. So that may change information here and there. Those are called mutations. Errors in DNA replication we call. Okay, you will see sometimes something like HIV is very difficult to make vaccine for. You know why? Because they make so many errors in writing. When they're synthesizing, they don't have DNA, but they have RNA. When they're synthesizing their RNA, they make too many mistakes. So majority of the times viral information changes. And even if you design an antigen or a vaccine for it, it will not work. But fortunately, our DNA doesn't make so many mistakes. It is very good at copying information. So very rarely mutations happen in the DNA replication process. Okay, right. So this is what we are doing now. We opened the DNA strand. We are using DNA polymerase. DNA polymerase is looking at the template, adding the nucleotides based on the template. Okay, how does it add the nucleotides? For example, in the template, if you see ATG, what will the DNA polymerase add? If ATGs are present, DNA polymerase will add what nucleotides? Will it add ATG or something else?
The counterpart.
Counterpart, exactly. That is complementary nucleotide is added. Remember in the DNA structure we talked about, for A, on the opposite side we'll have T, for G we will have C. So complementary nucleotide is added. If there is ATG, for A it will add T. If there is G, it will add C. For G it will add CR, depending on ATG. For T it will add A, for G it will add C. So complementary nucleotide is added. That is how DNA is synthesized by DNA polymerase. Are you with me so far? Okay. Before adding more information, I would like to show you that video till what we discussed. So you are clear about what it is, and you add a template in your head so that you can actually imagine further. Okay, now I'm going to show you a video. Okay, some basics that we talked about are repeated in these videos because videos are made to people to follow it. So some information is revised here, but I've taken small videos. They're just 3.5 minutes videos. They're not huge videos, just enough information that we need for our class. Okay, let's actually watch it till what I talked about so far. See if you could recollect that information.
DNA is a molecule made up of two strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The two strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand. And wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The two strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called Helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called Primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA Polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end.
Okay, let's stop it here. So why? Because a couple of things that we see DNA polymerase is now adding the nucleotides. I gave a simplified picture of this, but now you see more details given in this. First of all, they said that after separation of DNA strands happened, templates opened up for copying. So what we have seen, DNA polymerase itself cannot add nucleotides directly. It's like imagine a situation where a person who is building the wall, he cannot start himself. He needs someone else to start the first line of the bricks. Then he can only add the bricks to the existing bricks. He only join the bricks in a way. So like that, this video you have seen that something called primase, another enzyme. Okay. Here you don't have to too much focus on the enzyme, but just know the concept. The concept here is that a small stretch of nucleotides have to be present. Unless this nucleotide stretch is made, which is called a primer, because it primes the process of DNA replication, DNA polymerase cannot add new nucleotides. So unless there is stretch of nucleotides, meaning we have opened the DNA strands, like if I have to continue with the previous example that we have A, T, G, what has to happen before DNA polymerase begins the job? It has an assistant imager called primase. What does primase do? Primase first adds a few pieces. It will add T for A, it will add G for C, like that. It will make few nucleotides being added. So this is a job of primer. This is a job of primer. Primer is a stretch of nucleotides. Very, very important term in molecular diagnostics. Again, even in the PCR, primer is a defining factor. Primer will be different for COVID test. Primer is different for TB test. You use the same machine with different primers. Okay, so primer defining factor. Only when primer means here, DNA polymerase now will add, because primer is now fused nucleotides, right? Maybe 10 nucleotides approximately. DNA polymerase will now add the next nucleotide by looking into the template. So to begin the replication process, action of primase enzyme, which creates primer. Primer is the complementary sequence to the template strand. Only once the primer is added, DNA polymerase can extend that strand. Okay, remembering this information is too much, but remembering the concept is easy, correct? If you forget about primase, primer, what do you say? Before DNA polymerase begins, few nucleotides have to be added first. Then DNA polymerase can extend it. Now you understand this information, then you add more. Like I said, what is this called? What is the sequence called? This is called primer. Who is creating this sequence? Enzyme called primase. Okay. Now write down just the terms and the steps. And once you go through that, revise yourself, get familiarized, watch the video once again, then this information should be cleared. Okay. Are you with me so far? Can we add more information to this? Do you want to see this first part again? Or are you okay just to continue from here?
Okay to continue.
Great. Okay. So right. So where are we now? Where are we stuck? DNA strands are opened. DNA polymerase, first with the help of primase. Primer is added. Now we are adding nucleotides. Also in the video, we have seen they talked about DNA strands run in opposite directions. They also use the word something called 3', 5'. They're showing two numbers. Basically, they're talking about two ends of a DNA. This is very important to understand the next part. So let me first explain you what are the two ends of a DNA. Okay. So for that, let me take you back to the PowerPoint slide. So in this DNA structure, right, or something that we have already seen in the previous slides, right, and something like this. We talked about this is a double-stranded DNA. There are two strands. If you actually look into each strand, you notice that this DNA strand runs with a different polarity. What do I mean by that? If you watch this in this extended image, do you see how these molecules are organized? Do you see the orientation of this molecule called sugar here and here? Is the orientation same on both of them? No, it's flipped, right? It is flipped. So meaning it's like this. If you have some orientation to the DNA, one strand runs in forward direction, another one runs in reverse direction or backward direction. So this is what you notice. One of them is actually running like this, with this tip of the sugar is facing towards the upper side. Here it is facing downward. This is because they are actually oriented not like this, but they're oriented like this. The structures in them, they have orientation. The orientation is opposite in opposite DNA strands. This is what is called the polarity of DNA. Two strands run in opposite directions. One runs upward, another one runs downward. This is just a general picture. But to be specific, they give some polarity. They give some numbers. They say this is running in 5' direction. This is running in 3' direction. So where are these magic numbers came from? Why only 3, 5? Are they favorite to scientists? So there must be a reason why they are referring to 3 and 5 numbers only, right? That is because of the position of this. Okay, for this, I will quickly go to the next slide that we were looking into. Okay, I have written here this DNA strand. If it opens up, this is opened up, right? This strand opened up. It is running from 3' to 5'. And the second strand, opposite strand, should run from 5' to 3'. Okay, so it is 3 to 5 and it is running from 5 to 3. Okay, look at this. So to be specific, what we are seeing, this is a structure. So look at the 3 and 5 are highlighted here. This is just another image of the same structure that we have seen before. This is a sugar running in this direction, downward direction, and this sugar running in the upward direction. So what you notice here, this 3 and 5 are labeled here. This to the sugar, 5' side is where phosphate is added, because nucleotide has 3 parts, right? Sugar, base, phosphate. To the sugar is the anchoring molecule. Sugar is what is bound to phosphate, what is bound to base. So on the 5' side, because they normally number like 1, 2, so they begin somewhere here, like 1 here, 2, 3, 4, and 5. These molecules are numbered. On the 5' side, there is phosphate. On the 3' side, the third molecule, third carbon, will have another group called hydroxyl group, OH group. OH group. So third carbon on the sugar has OH group. Fifth carbon on the sugar has phosphate attached to it. So there are two sides of the same sugar. One side phosphate is added, another side hydroxyl group, OH group is added. So the polarity is defined on this side. You see it here. Which one is to the bottom? 3' side or 5' side on the left hand side of this DNA strand? Is 3 at the bottom or 5 at the bottom? In the structure of this molecule, which is at the bottom? So 3 is at the bottom. 5 is here. Again, another 3 is added to the 5. Then 5. So basically what happens, if you have a molecule like this with the 3' end at the bottom, the 5' side is added to the next molecule's 3' side. Because they're phased like this. 3' side is phased like this. So every time bottom we have 3' side and 5' is on the top. This 5' joins to the next incoming nucleotide's 3' side. Like that, 5 is added to 3 and next 5 is added to 3. The bond forms between these sides. Bond forms between 5' side to the 3' side. And the molecule is running from 3' to 5' because you know the bottom is 3. The last part of this is ending with 5. So we say this is 3' side, this is 5' side. Even if you absorbed 70% of this information is all right. So what are we learning here? DNA strands don't run in the same way. They have polarity. If one runs from the 3' to 5', another one runs from the 5' to 3'. Opposite is true. That is what you see here. Here this is actually 3' is placed at the bottom. On the opposite strand, 5' is placed at the bottom. Same thing is true here. 5' is placed at the top here and 3' is placed at the top here. So that is why it's called 3' side and 5' side. So only what you need to remember, DNA runs in opposite directions. If there is a 3' side on one side, 5' side will exist on the other side. And same vice versa happens on the other end of the DNA strand or DNA template. Why is that important? Here the reason why I had to introduce this concept is this has little bit of variation that it creates on how each strand is copied. Okay, so far are you with me on this? Did you understand what I just talked to you? Orientation, have you understood now? And why the numbers 3 and 5? Okay.
So just one thing, sir.
Sure.
Sir, both 3 and 5' are connected to the phosphate group, right? You said that 3' is connected to hydroxyl group, sir. I didn't understand that. It looks like both are connected to the phosphate from the diagram.
Correct. Good. Very good question. So when phosphate is added, like for example, we'll take this phosphate. This phosphate is added to the hydroxyl group of this. It's a chemical reaction. This 3' will have OH group with which phosphate actually interacts with. You make a bond. This is called a phosphodiester bond. Bond between nucleotides. A glue between each nucleotide comes in the form of phosphodiester bond. That OH I was telling you is what actually originally present on the 3' side, but when phosphates reacts with it, now it creates a bond called phosphodiester bond. So what is interacting here? Phosphate of the 5' side of the sugar to the OH group of the 3' side of it, thereby joining the phosphate to the OH. The bond forms between phosphate and the OH. Okay, but to define, we say this is 3' hydroxyl group, this is 5' phosphate group, we say.
So is the bond with the phosphate is with the carbon or with the OH group?
OH, not carbon. Carbon is here. Okay. To the carbon, we have functional groups. I don't know if you have any background in chemistry, bit of chemistry. So it doesn't directly bond with C. It bonds with the functional group attached to it, which is OH group. It's like a hydrolysis process. You have OH. PO4 minus comes in the picture. You get H2O removed here. Then you form a bond between them. Okay, I think that's enough. We don't need to get into more details. Otherwise, I can show you exactly the bond formation, which is too much for us. Okay, but if you're interested, you can look for that, how this bond is formed with the help of OH. PO4 minus and OH, they join together. Okay, why is that important? Because DNA polymerase is trying to add nucleotides to each other. When it is adding nucleotide, it means that each nucleotide is joined between 3' and 5' side with the phosphate group, resulting in phosphodiester bond. Okay, phosphodiester bond, right? So that can make a difference here. Imagine if the strand is running in opposite sides. This template here is 3' to 5'. This template here is 5' to 3'. And imagine that you are extending the DNA strand with the help of DNA polymerase. Okay, you opened it and you also notice that DNA has not completely opened. You didn't separate whole DNA into two parts. Only in the region where you are making the new copy, only in that particular location. So locally, if I have to say, it got separated. And DNA polymerase is starting to act on each side. But the synthesis of DNA happens in what direction? If you really understood what I've explained, you should be able to answer this. In what direction should DNA get synthesized? Template is either in the 3' to 5' side. Here it is in the 5' to 3' side. But in only one direction, you create phosphodiester bond. What is the direction? I'm taking you to the image I was just showing you. In what direction phosphate or phosphodiester bond is added? 3 to 5 or 5 to 3? In what direction extension happens?
3 to 5.
3 to 5.
5 to 3.
5 to 3. Okay, right. It needs a bit of explanation, but actually what happens here, template strand is always 3 to 5. Template is always 3 to 5. And what I was telling you, complementary nucleotide is added. Every time DNA polymerase adds, it looks at the template. Template as A, it adds T. Template as Z, it adds C. Okay, and I was telling you both the DNA strands run in opposite direction. Actually, you are creating the opposite strand to the DNA. So it has to run in the opposite manner. If your template is always 3 to 5, your product is always 5 to 3. So if this new strand have to run because this is the existing strand. Let's say you are creating a copy of this. You know what happens? This strand you begin with 5' side. You begin with the 5' side because that is where phosphate is. And this 5' end of the previous nucleotide will be added to the 3' end of the second nucleotide and vice versa. It continues like that. 5' phosphate is added to 3'. Again, this 5' phosphate is added to 3'. So it runs from 5 to the 3' in the opposite strand. Although we are looking into the template strand, this is template, right? So in a way, when you are creating the strand, new strand, it should look like this. Because you started with 3, but on the opposite side it will be 5. So you make the 5' side here, and then 3' side will be here. And next, what you do, you bring in the new nucleotide, so you join them. Opposite to what you are seeing in this, which is the case with this DNA strand. I will show you in the video. This may become a bit clearer, but I'll repeat what I just talked to you. Always template strand is 3' to 5' strand. So this strand is the template strand, always. Or this side of the template will act like a template strand. So 3' to 5' direction. Template orientation is always, let me make a note of this. Template strand is always in the 3' to 5' orientation. Template is always in the 3 to 5' orientation. And the product is always in the newly made DNA is always in what direction? Opposite direction, which is 5' to 3'. Just remember these two and imagine the rest of the things as I've explained should make sense to you. Even if you forget this, remember this actual information. Template always runs from 3' to 5'. Product runs from 5' to 3' because they have to be opposite to each other. Okay. Okay. So that is what happens. That you notice now here, we are adding more information from the slide. So this is 3' to 5' strand. 3' to 5' strand. This is acting like a template because you can add a nucleotide here with 5' attaching to the next nucleotide, incoming nucleotide. So 5' to 3' DNA can be made here and continuously made. Why? Because it doesn't have to change the direction. Template strand is actually rightly oriented in this DNA strand, the top strand. The stop template strand, we have the DNA template in the 3' to 5' direction. So what happens here? The newly added DNA strand will be made from 5' to 3' without any disturbance. It can continue to add by unwinding the DNA. It will keep adding the nucleotides. It doesn't have to stop or pause. But it is not true with the opposite strand. Because how is opposite strand situated? Opposite strand, which is opened up like this. I was telling you the top one is 3' to 5'. Bottom one is 5' to 3'. In what direction nucleotides have to be added? Is DNA polymerase oriented towards this side or towards this side?
Yes, sir.
Towards the end, right?
Yes.
It's actually synthesizing to backwards in a way. So it is sitting here, the DNA polymerase is sitting here and going towards the end. But in this strand, in the top strand, DNA polymer is going towards the remaining portion of the DNA strand. So it can continue, it doesn't have to reorient it. It will continue to add nucleotides as it sees the template. But there is a problem with this DNA strand. Why? Because DNA polymerase has to run from the 5' to 3' direction. So every time it is starting at the tip at the end and going towards the end. But it hit the end. So what it has to do now? It has to again go back. So it's like it's actually adding bits and pieces. It is adding a piece here. Now again, DNA template opens up more. So this DNA polymerase again gets shifted to the next location. So like that, every time it is actually joining few fragments. It is only creating few fragments in one strand. The other strand, it is actually constantly moving into the DNA because as DNA is opening up, it is getting access to more and more template and it is now adding nucleotides. That DNA strand is called leading strand. This is called leading because it is continuously, it is able to synthesize the DNA. So now I'm introducing two terms here. One is leading strand. What is leading strand? The strand of the DNA where DNA polymer is able to continuously synthesize it with no interruptions. Whereas lagging strand, can you explain, can you guess what is a lagging strand?
Which has to stop in between.
It only is creating fragments each time. It is only copying this portion of the DNA because it has to, there is a rule that it has to follow, that it has to go in the 5' to 3' direction in synthesis. So here what is happening? It is going in that opposite, backwards. But every time when the DNA template opens up, it has to go back and again synthesize. So meaning it's actually synthesizing fragments of it. This strand is called a lagging strand. And the fragments that are created here are called Okazaki fragments. Okazaki. So it is creating fragments. They gave a name to these fragments. And these fragments are called Okazaki fragments. Okazaki. Just know this. Know that there's something called fragments. They have a name to it. That is Okazaki fragments. Why DNA synthesized in fragments on one strand? Because of the requirement to satisfy the orientation. Okay, we are coming to the last part of the replication. We more or less figured out majority of the information now. We're coming to the last part of it. If you understand leading lagging strand, this is a key aspect. For that, I have to explain you the orientation. Only when I explain you orientation, I can tell you why one is leading, one is lagging. Do you see the need for information here? Need to tell you about 3' and 5' directionality here?
Sir, I just want to know Okazaki. What is it like? I just forgot.
Okazaki is a fragment, name of the fragments. Because as I was telling you, on the lagging strand, it was creating fragments each time. Only small strand and again going back, creating another strand as template is opening up. Each fragment is called Okazaki fragment. The name of that fragment is Okazaki. Because probably scientists called with the name Okazaki might have recognized it. That's why it goes with that name.
Sir, won't the lagging strand, once it starts, continue till the end, till 5?
Won't the lagging strand continue till the end? Okay, so I think two things. First of all, I told you DNA template doesn't completely open up. Imagine that DNA is like as long as my hands. Okay, imagine like that. My arms are twisted. Imagine that they are twisted. So what happens? Only here it opens up. Only this portion opens up. So the lagging strand is only accessible to till whatever part is opened up. In this structure, it only opened up till here, right? So DNA polymerase sits here, begin to add the nucleotides in this opposite direction. Then what happens? We still have to replicate the rest of the DNA. This is only a small portion of the DNA. Then remaining DNA opens up. Then this DNA polymer has access to now, let's say this part. The next remaining part further got opened. So what has to DNA polymerase do here? It has to go back till it opens up and start synthesizing from there. Primase goes there, add the primer. DNA polymerase goes there, adds nucleotides to the primer. But the same thing is not required in this strand because the leading strand, DNA polymerase hit the end here, let's imagine. But now again, DNA polymerase, the DNA opened up further. So what happened? It can continue to add nucleotides without the need for adding new primer. But here, each time primer has to be added till the amount of DNA that is available from the template is what read by the DNA polymerase to add new nucleotides. Is it making sense?
Sir, yeah. One more question is, wherever we in the picture we have a lagging strand, then towards the right, if it moves till 5, won't it be complete because it has access to this part?
Okay, come again. Can you repeat the question?
Wherever it is written lagging strand.
This is lagging strand, okay?
Yeah. So the formation of complementary part. Once it starts from here, since the strand has already opened up,
so
won't it? Yeah.
Right. So it has access here. Okay.
Yeah. So won't it continue till the last part? That is where it is written.
So basically, it starts here. It finishes till here.
Okay. Okay.
But it's only small portion of the whole DNA. We still have this much amount of DNA to be copied, right? So what it did, it started only at the end and copied the last part of the DNA till it opened up. But we still have so much of DNA that is left out to be copied. So you open up more DNA and again repeat the same process. Primer is added and DNA polymerase extends.
So let's say if it opens more.
It opens, okay.
Yeah. And it again gets access to primer at some part. And then again it's moved towards the right in the lower band. Won't it reach the already existing part?
No. It reaches the already existing part, but remember, it is beginning with the primer.
Okay, so it won't be continuous.
It won't be continuous. It actually will result in the gap there. It will go and hit the primer, but no joining happens. It won't be able to join here. So this will have a bit left out. So fragments. So lagging strand will be created as the fragments with the gaps in between. Your question actually, you will get the answer to this because something else has to be done to the lagging strand to complete it, to fill the gaps. I think now let's watch the complete video. You will get this whole information and see if you're still stuck with that concept, we'll try to fix it. I think your answers will be cleared, the questions will be cleared from the remaining part of the video. Okay, because it's easier to see it than just me explaining you with the bits and pieces like this. Now let's finish seeing the video. Okay, I'll begin slightly before so that you can follow the information that we are looking into.
The two strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand. And wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The two strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called Helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called Primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
Okay, is that clear to you? The steps that I talked about in this? Okay, so what they talked about here, rest of the information is this. So we have leading strand and lagging strand. In the leading strand, DNA polymerase, as the template opens up, it continuously will add the nucleotides, creating no gaps there. So it is continuous strand. And then we have the lagging strand where, given the orientation requirement, as the DNA template opens up, new primer is added, and to the new primer, DNA polymerase keeps extending it by adding nucleotides as defined by the template. Like the two strands, one continuous, one discontinuous strands are made here. Okay, one continuously made and the discontinuous. And other enzymes they talked about, which you don't, if you don't want to remember, that's all right. But is what we have to remember here, that is primers have to be removed because primers are RNA primers, they're not DNA. So another enzyme called exonuclease comes into the picture, it removes those primers. So then to fill that gaps, another DNA polymerase comes into the picture, but don't have to specify that. Just say that DNA polymerase will fill the gaps. Then we have those, between the fragments, we have a gaps left out, right? An enzyme called ligase, which we are going to talk about in 9.1. This is an enzyme like a gluing enzyme for DNA. What does it do? It will fix the gap. What is the gap here? No phosphodiester bond is formed between the fragments. So this enzyme will come and fix the gap by adding that phosphodiester bond, thereby resulting in two DNA strands. Now they look as if they are continuous strands, but it is made as fragments in this and it is made as a continuous strand in this. This is how new strand is made differently for two template strands. But end of the process, two DNA strands are copied, creating how many new strands? Two more new DNA strands. The old one will have one new one, and the second one, old one, will have another new one. So basically, that is why they call it a semi-conservative mode of replication. So why? Because each product will have one old DNA, one new DNA. Because template and the newly produced DNA. What happens towards the end? The new DNA will be formed like this. So old DNA opened up, old and old ones, two old copies opened up, right? New one is formed here. This will pair with this. So they both get separated and this will pair with this. So each one will have one old DNA strand, one newly synthesized DNA strand. That is, it's called semi-conservative. Semi, partially conserved, meaning partially old one is still present in them. This method is called semi-conservative DNA replication. Even NCERT class 12th book, they talked about these experiments, which we don't have to get into, but this is how DNA replication process happens. Okay, briefly, I'll review the process with highlighting the key terms. And here I'll take the help of the document I have posted on the group. Okay, the outline that I have given you, let's have a look at the outline. So this is just to make sure that you understood what is required here. That is why I made this outline. So did you understand when and why does replication happen? When does replication happen?
S phase.
synthesis phase before cell division, the interphase. Very good. Why does it happen? Anyone else? Why does it happen?
Cell division.
So to satisfy the cell division, to maintain the DNA number, to maintain the chromosome number, right? If no copying happens, DNA becomes half each time. We don't want that. We want every cell to carry all the instructions. We want 2 copies that we received from our parents. So DNA has to replicate every time before cell division happens. So it happens during the interphase, right? Okay. Enzymes. Do you recognize that there are several enzymes required for this process to happen? Do you remember any names? Can you recollect few names of enzymes? The most important enzyme in the process, which carries out the majority of the process here?
DNA polymerase.
DNA polymerase, enzyme that creates the polymer of these nucleotides. Okay. Which enzyme opens up the DNA? Anyone else? Which enzyme? The enzyme that opens up the DNA, unwinds it.
DNA helicase.
DNA helicase, very good. Okay, right. And the enzyme, even before polymerase acts, it needs to synthesize few stretch of nucleotides. How are these stretch of nucleotides called?
Primer.
Primer. Why is it called primer?
Primase.
Priming means starting or initiating the reaction. It's called a primer because it's priming the process. Okay, it's a primer. The enzyme that creates a primer is?
Primase.
Primase. You see the connections? You make the connections. You may not remember them, but you make the connections and write down in the notes. You will remember it. When you recollect it, you will have the clear picture again. Even after prelims, once I think everything kind of goes down in your head, when you try to pull up the information, when you read your notes, it should become clear. Okay, right. So we talked about helicase, DNA polymerase, primase. Okay, the rest of the enzymes, I just leave it. You don't have to remember, but just to repeat for yourself, exonuclease. Nuclease is something that removes or degrades the nucleotides. So exonuclease is to remove those primers. Okay, and the one that joins, it is important because we anyway have to come across this again in biotechnology. So the enzyme that actually joins the DNA fragments is called
Ligase.
Ligase. Restriction endonuclease or restriction enzymes or molecular scissors, ligases are the key tools in constructing new DNA in biotechnology. Okay, we'll come back to this ligase later in coming classes, but these are the enzymes. Okay, now you recognize it. Do you recognize leading strand and lagging strand?
Yes.
What is leading strand?
One that forms strands continuously.
One that forms the DNA continuously. Okay, what about lagging strand?
That creates fragmented strands.
One that forms a strand in the form of fragments or fragmented strand is made. Why is that? What is the reason behind it? Why is leading strand is continuous, lagging strand is discontinuous?
Because the DNA structure opens up towards the leading strand.
Okay, that is one aspect of it, but they should still go back. They should actually still work like that. They can go in the direction towards where the template is opening up. Right. So it is not exactly because template is only opening partly initially, but also in addition to that to match something else. What is that requirement?
It has to form from 5 days to 3 days. Right?
Correct. So template strand orientation is what? Template strand orientation has to be from
5 to 3.
3 to 5.
3 to 5.
Template is 3 to 5. Remember, small number to large number. Template has to be 3 to 5. The newly formed one will be opposite. 5 to 3. So that is how you remember it. The template which is already existing has to run from 3 to 5, lowest number to highest number. Whereas the opposite strand, in the opposite direction, newly made strand has to be created. So newly made strand is 5 to 3. So to satisfy the 5 to 3 synthesis, as only part of it is opened up, leading strand can continue, lagging strand cannot continue. It has to discontinuously make the new strand. Okay, right. So you know all these terms. Template DNA. What is template DNA? Writing, I'll let you write. So next we'll go through the terms, see if you understood all these terms. What is template DNA?
Each of the separated strands.
So meaning existing DNA, the original DNA is called template because it is guiding the newly forming DNA, right? So template DNA is the originally existing DNA with the instructions that we received from the parents. Okay, that is template. Something that is guiding the process of DNA replication. Helicase, I think we already talked about. DNA polymerase, we talked about. I didn't specify this replication fork or replication bubble, but did you figure it out by watching the video?
Yes.
So why is it called fork? And what is fork here?
When the helicase opens up, then the whole open
Correct.
structure
It looks like a fork because it opens up like this. This is called fork. This is called a bubble. When? Because it continues further, you know what happens? While it is continuing, renaturation happens towards the back. So at one point of time, it will look like it's a bubble in the center. Okay, so this structure, this appearance is called fork, replication fork or replication bubble. If it is only opened up in the middle, both the sides ends are actually closed. Okay, primers. Can you give me a short sentence on primers?
It is a it is made up of pieces of
Sorry.
It is made up of pieces of RNA by the enzyme called primase.
Correct. Okay. So this is complementary nucleotides. Okay, to begin the or to initiate the or to prime the DNA synthesis process. Okay, in this process, so this is what exactly used in PCRs as well, the machines where DNA is multiplied. So we can artificially do it. This was one UPSC prelims question. Year or before year, they asked this because of the advancements in DNA technology, which of the following can be done? We can synthesize the DNA outside the cell. That is the PCR reaction. So this part is also useful for solving some questions from biotechnology in the prelims paper as well. Okay, so primers are those few stretches of nucleotides. Leading strand, lagging strand, we already talked about, right? Semi-conservative DNA. What is semi-conservative DNA?
Where a part of the old DNA is conserved in the new DNA.
Correct. So semi means the newly formed DNA containing one old strand, one new strand. So semi, partially conserved. Scientists had to do experiments. How do they know? We're actually talking and struggling to understand this concept. But without able to see DNA, you cannot see with your eyes. Even we need more advanced microscopes than electron microscopes. They could actually figure this out with clever experiments to show that indeed every newly formed DNA has one old strand, one new strand. NCERT books talk about this type of experiments, class 12th.
Sir, I have a question, sir.
Go ahead.
Sir, template DNA is the unwinded part of the DNA? So I'm confused between the primers and template DNA. So you call the primer also the template, right? So that is
No, I'm not calling. So this is primer. This is the template. Primer is what is bound, what is attached to the template in opposition to it. So template primers are primer is in a way product, but to initiate the product, primer has to be made by another enzyme, attach it to the template. This is a template. This is primer.
Template is the original structure, part of original structure.
Template is the original DNA. Yes.
And so primer, we can define it as something like which starts the process.
Yes, you can say it is something that starts it, but it is being used for something else as well. That is why they say primer is a complementary DNA sequence to the template. It is complementary to the template, and we don't have big DNA here, only few letters added here. It's very small. It's usually 10 nucleotides. So about 10 nucleotides.
Okay, thank you.
Okay, right. Anything else?
So leading and lagging are basically the new strands that are added. The new ones, or we can
template. Leading is the template strand one side.
On the top.
The lagging is opposite side. Continuous, discontinuous strands are the products. The one that is leading the process, the one that is lagging the process. So it is referring to template. Products are called continuous, discontinuous.
Is leading always top strand, or just for the sake of discussion did we use top?
It can be any. Only I have to show in one orientation, but it could be top, bottom, doesn't matter. The one with 3 to 5 prime. The one
so leading is always 3 to 5.
Leading is always 3 to 5. And 5 to 3 is the lagging. Okay, good.
Sir, one question. The leading is formed continuously and the lagging is formed in parts. So is there a time gap between them for the ultimate cell, I mean, the duplicate?
To my knowledge, both has to happen at the same rate, because unless whole DNA is made, cell cannot continue because they have something called checkpoints just to ensure that this is done. So maybe lagging strand might delay the whole process because it may pause the whole process for a bit, right? But overall, the rate at which it happens is very, very fast. We're talking about making copy of the DNA within 23 hours in our cell. And where majority might go for the DNA synthesis itself, so it may take few hours. And here we are writing 3 billion letters. We are copying 3 billion letters. Imagine how fast it can be. I think if I'm not wrong, if I have to guess, because I forgot the actual number, somewhere around 1000 kilobases per second, I think it copies. So it's much faster than our typing. Anyone who can imagine the fastest typing that is possible. Okay, but overall, so the delay may happen because of the lagging strand, but not as much as we can that notice or delay the whole process. Okay, does it answer your question?
Yes, sir.
Okay, should we go ahead now? This is about DNA replication. In the hand, in the outline I've given to you, I've given the link for the video as well. So watch that video if you want to repeat this information later. Now we'll move on to the last topic in this unit. Okay, now we finished talking about making copies of DNA, DNA replication, DNA doubling, DNA duplication. Any of these terms are referred to this. Duplicating DNA, replicating DNA, doubling DNA. Okay, good. And one thing you have to remember, which we may extend later, errors can happen. This DNA polymerase can make errors sometimes. So errors, as far as they don't make too much of a problem to us, our body has a repair system to fix it. This you will study where you probably heard about CRISPR-Cas9 system, CRISPR-Cas9 genome editing tools. There you will learn about this type of molecules coming into the picture, fixing the mistakes that DNA polymerase does, but while editing the problem. Okay, inside our body, we have this DNA polymerase has a mechanism to proofread it. It even goes back and checks whether it added right. And proofreading also happens before it proceeds to the cell division. So imagine how nicely this is organized. It copies it with the help of many enzymes. It even proofreads it and fixes the problem. So usually we are not left with too many mistakes, but rarely, very, very few nucleotides are by mistakenly added or by mistakenly kept. Very rarely that happens. Otherwise, imagine we might have gone through several cancers in our lifetime. But that's not the case. It is made more or less foolproof. But that's not the case with viruses. Viruses make enough mistakes, which doesn't cause their lives, but it causes problem to us. Because if you make a vaccine for it, vaccine will not work because the information is changed. The information towards which you created the vaccine is changed now. So drugs may not work forever. Vaccines may not work forever. For vaccines, which adds more mutation, more, excuse me, more errors that they incorporate. Sorry.
Sir, is it that the lagging strand has more errors than the leading strand? I mean, because this leading strand, lagging strand has more, intermediate steps.
Okay. Your question is whether leading strand will have more errors than the lagging strand.
No, the lagging strand will have more errors than the leading strand because lagging strand has intermediate processes.
But still, do you think what type of function the DNA polymerase changes here? There are two possibilities. You can argue otherwise. You say in the lagging strand, DNA polymerase is more meticulous because it gets to do just bits and parts of the work. Whereas in the lagging strand, it has to continuously do the job, make more mistakes.
Yeah.
I don't know. Actually, I have to check whether there are more errors, which I've actually not thought about. Okay, I don't know the answer to it. I may have to figure this out. But I doubt that if there are going to be more. So it could be someone who have thought about it and observed it. So let me make a note of that.
But then how one will differentiate that, because ultimately it will be a part of some other cell. So there's
in the process, you have to pause it. We have technology where we can actually freeze it in the process. something called cryo-EM microscopy, Nobel Prize winning research. There, scientists can freeze the process in between and see in the process before they fixed it, is there anything like that? Okay. But I appreciate that you're able to think beyond what I'm teaching. That is most important. That tells me that you're following my class. Okay, that's great. Okay, right. See if at all if someone thought about it and figured it out, just for curiosity. But exam, you don't need that information. Great, guys. Now we are moving to the last part, which is protein synthesis. Okay, so what is protein synthesis? How does it happen? So DNA replication is a different concept. DNA replication is where we talked about replicating DNA, but protein synthesis is actually reading the DNA, converting information in the DNA into proteins, right? So I'm connecting back to the big picture here. So what we receive from our parents is instructions in the form of DNA. But DNA cannot be everything because DNA is only the storage part of the instructions. It always sits inside the nucleus storing the information. And it never ever leaves the nucleus of the cell. But still, cell has to give instructions to every other process that is happening in the cell. So nucleus is only a small part of it. There is cytoplasm outside the nucleus where a lot of actions are taking place. For that, you have to give instructions, and these instructions have to come from DNA, meaning somehow this information from the DNA has to leave the nucleus and go out. Correct? Okay, that is one aspect that you need to know. Second aspect that we need to understand here is DNA cannot leave. Something else has to go carry the information. That something is RNA. That something is RNA. RNA is very much like DNA with few differences from DNA. We call DNA as a deoxyribonucleic acid, deoxyribo, and RNA is just the ribonucleic acid. It is missing the deoxy part. That is because structurally they are different. Sugar in DNA, it doesn't have oxygen, so it is called deoxy. Ribo has ribose in it. So the sugars are different. Between DNA and RNA, what is different? Between DNA and RNA, DNA contains what is called deoxyribose sugar. RNA contains what is called ribose sugar. But they both are otherwise very much similar. They both are made up of nucleotides. They both are made up of ATGCs, except in RNA, instead of ATGCs are common because DNA contains ATGCs. DNA contains ATGCs, write it here. So if this is DNA we're talking about, which is made up of nucleotides. Okay, what nucleotides is it made up of? A, T, G, C. But RNA is also made up of nucleotides, but instead of T, RNA contains U. So it contains U. So except this, remaining three letters are common to DNA and RNA. Okay. The next point I would like to tell you as a difference between DNA and RNA. DNA is double helical structure that we're watching it. You've seen it multiple times. It's a double helix. Is RNA also double helix? No. RNA is a single-stranded DNA. It only has one strand. Viruses may contain RNA with extra strand. Some viruses have double-stranded RNA, but usually inside our body, we always see single strand of RNA. So the last point here, DNA, RNA differ in terms of their function. What is the function of DNA? DNA is a storage material. It stores the instructions that we receive from our parents. Correct? What about RNA? What are the functions of RNA?
Transfers information.
It takes the information from the nucleus to the cytoplasm. It takes the information, it makes a copy of exactly how we talked about, more or less, not exactly, but similar to what we talked about in DNA replication. In DNA replication, a copy of the DNA is made. Here, a copy of the DNA is made in the form of RNA. So following this, not T, but U as an ingredient with remaining AGCs. Okay, so it has multiple functions, not one. One, you said it carries the information, it carries the message or carries the information. This is one. The two more I'm going to introduce as part of the next discussion. So for now, just remember RNA performs variety of functions, unlike DNA. What are they? One of them is carry the message from the nucleus to the cytoplasm. Okay. First, I'll show you a brief picture about it, brief idea, then I'll add details to it. So this information from nucleus to the cytoplasm happens in a process called protein synthesis. The title here is protein synthesis. Information from DNA first gets copied onto RNA. So DNA is present in the nucleus, right? A copy of this is made into RNA. This specific RNA, which is acting like a messenger, is called a messenger RNA. This RNA is known as mRNA. M stands for messenger, messenger RNA. Messenger RNA. Because it is carrying the message from the nucleus to the cytoplasm. And what is the ultimate goal with this? Ultimate goal is based on the instructions in the DNA to make protein, to create protein. Because proteins are the working molecules. DNA is a storage molecule, protein is a working molecule. So based on the instructions in the DNA, you create proteins. But these proteins cannot be made in the nucleus. Why? Because the machines to synthesize proteins, which are ribosomes. Have you heard the term ribosomes? Ribosomes, these are molecules found inside our cells. So these are the machines to synthesize proteins. So those machines are not present inside the nucleus, but outside the nucleus. So message has to leave the nucleus to reach ribosomes. And this comes in the form of mRNA or messenger RNA. Once it leaves this, it goes looking for ribosomes, and ribosomes what reads the information in the mRNA. So it uses mRNA as the template and creates proteins. Okay, I'm providing an overview. I'll give the details, then you have to add more information to each step. So now the first part, just understand what it is. Understanding is more important for this part. Then we'll add details. Then while adding details, write down the information. So what did I tell you so far? Protein synthesis is a process where DNA, which is a storage material which carries instructions, a copy of it is made in the form of mRNA. And this process has a name to it. We call this step as transcription. This is called as transcription. This is the first step in protein synthesis. Is the transcription. What does transcription mean? One prelims question, This was a UPSC prelims question, I think 2 years ago. This came in the exam. They asked about process called transcriptomics in biotechnology refers to which of the following? Transcriptomics is a process of understanding RNA, studying RNA, mRNA, most important. Because transcription results in mRNA synthesis. Our transcription is the process in which mRNA is made or synthesized. So these terms are important for both prelims and mains. But the second part, where mRNA is read by ribosomes to create proteins, is called translation. It is called translation. Why is it called translation? Can you relate anything else to this? For both transcription and translation, you can relate to remember it well. Transcription first. Have you heard about medical transcription? The term medical transcription. Some people who actually read the code language that doctors write or they type, so they don't have time. And there are people, there's a dedicated market being created for this where they can read the short-term language of them and expand it and write a summary of what doctors are trying to say. This is decoding what doctors are saying. So transcription is decoding the information from DNA into RNA. So it's called transcription as used in medical transcription, where medical transcriptionists convert the information from what doctors say into more detailed document. They're decoding what doctors are saying because it's a big challenge, right? Decoding what doctors normally write. So medical transcription is special.
So there is this post called Steno in administration who does this shorthand writing of whatever the those.
Exactly.
Is it the same like the medical transcript?
Medical transcription, what do they do? So doctors actually either scribble on it or they actually record it. What do they do? I remember because I lived in Canada where I've seen that. As soon as I met doctor a couple of times, they said they go home, they go back to the office and they record what they just said. So either audio or the short version of it in the medical terms is given to the someone else, which is expensive because those stenos are expensive. You have to pay them more. But if you send them to countries like India, there are many people who can work for 5,000, 10,000 rupees and convert this information with a background in bio or training in bio, give a detailed document of what doctor said, send it back so they can keep the record of that information. Okay, it's a specialized field that has been created like this.
In more simple terms, the same process is use it for creating subtitles also for videos.
But subtitles is in a way, not subtitle, but transcription of it. Subtitle is where you only write the sub part of it. Transcription is where you transcribe what he's saying. For the video, writing the transcriptions, exactly.
Yeah.
Making the transcript of what they are saying in the video, exactly. Correct. So we can create our transcripts of this class in a way. Okay, next. Translation. What does translation mean? Literal terms.
Converting it into our language.
Another language. Let's say that you apply for admission in Germany, in a German university. What if they send you admission, the decision in German? What do you do? Nowadays we do use Google Translator, translate it. Right? Otherwise you have to go to a translator to translate it from Germany to English. So yeah, we are translating it from the language of DNA into the language of proteins. They are not same. DNA is written using nucleotides. Proteins are written using? Answer is here.
Nucleic acid.
Amino acids. Proteins are polymers of amino acids. DNA is a polymer of nucleotides. Proteins are polymer of amino acids. So they are the individual units here are amino acids. Whereas DNA is a polymer of nucleotides. They are written in languages. Different alphabets are used. So we need a translator called ribosome. So for us, Google Translator is ribosome. So this is the overall idea about the process. I'll just repeat what I just said and we add a little bit of more information to finish this topic. So we're talking about protein synthesis here. What is protein synthesis? The process that leads to the synthesis of proteins. So how are proteins made in our body? Proteins are made as per the instructions in DNA. As per the instructions in DNA. What happens here? DNA, which is acting like a storage material present in the nucleus, makes a copy of this information in a process called transcription. I repeat this. So for this, just write a flow chart, write a diagram rather than big sentences. Sentences just to make connections here. So you draw images like this if you want. Yeah, I have an image here. So this is a cell. So we have a nucleus here. So what happens from the DNA? By using the instructions in DNA, you create a copy of RNA in a process called transcription. So transcription is where information from DNA is copied onto mRNA or messenger RNA. It is copied onto messenger RNA. Follow this flow chart. Just write down the sentences where you want. Nucleus. This is happening in the nucleus where DNA got copied onto mRNA. Okay. What are we copying here? We're copying message. And you already have a knowledge about how message is copied. It exactly follows what we talked about in DNA replication. So look at this. You can actually figure it out from this information. So this is template strand. Template is always 3' to 5', correct? You always can create a new strand, whether DNA or RNA, always in the 5' to 3' direction. And mRNA is always double stranded or single stranded? Single stranded. So you only look at this. We only made one stranded nucleic acid here, which is RNA. And 3' to 5' is the template. Do you see how is the RNA made from the DNA here? There is a T here. What is being added here? A. There is A here. What is being added here? It's supposed to be T, right? But there is no T concept in RNA. Instead of T, we have U, uracil. There is uracil in place of T. So opposite to A, they added U. Similarly, opposite to C, G being added, opposite to G, C being added. This is how RNA is made. This is how decoding works. You read the letter, complement letter is added. If it is RNA, instead of T, you add U. Otherwise, you create this single-stranded RNA by looking at DNA. This is how every time a protein is made in your body, this is what it does. It goes to that part of the chromosome, and protein may be coded by a small portion of the DNA called a gene. Not every part of the DNA gives rise to the proteins, right? Only genes code for proteins. So we have to go to that portion of the chromosome called a gene. Remember the definition I gave in the previous classes about gene? Gene is that portion of the DNA or a nucleotide sequence which gets converted into protein or translated into proteins. Not everything on the DNA is genes, right? It's only bits and pieces here and there in the DNA. So that portion of the gene, let's say you're making insulin. You want to make insulin hormone, which is a protein hormone. So what you do here, you go to that portion of the instructions on the chromosome and you try to make an mRNA of that portion. So mRNA is much, much smaller than DNA. You have a huge length of DNA with 3 billion letters. mRNA is few thousand bases. So only that part, that switch, you actually turn on and create mRNA for that. As per the need. If your body needs hormone, insulin, it goes to that part and creates mRNA. So unlike the DNA replication where from the starting to end, you have to copy everything there. Here you don't copy everything. You only copy the portion that you are interested in that needs to be translated. These things, whatever videos you watch, whatever books you read, you won't get that idea. The reason why the teachers are important here is they actually kind of worked on these concepts and they can tell you this information. Okay, so right. So transcription results in mRNA. Now what happens is mRNA goes out. If I show you in the other video, look at this mRNA. It is leaving the nucleus. This blue color region is the nucleus. It is leaving the nucleus, comes out looking for ribosome. And this complex structure is a ribosome. Ribosome has two parts. Small units, big unit, we call it. This is a Nobel Prize winning research. Indian-born scientist received a Nobel Prize along with 2 other scientists. Anyone knows who is that scientist received a Nobel Prize for this in 2009? For discovering the structure and function of ribosomes? Venki Ramakrishnan, Venkataraman Ramakrishnan, or Venkateshan Ramakrishnan. Venki Ramakrishnan, Indian-born scientist from Tamil Nadu, still is a big critic for Indian research, Indian science research. He comes to Science Congress in India. He comes to IAS, Ministry of Sciences regularly, talks to students, encourages them. And he's a scientist who received a Nobel Prize along with one American scientist and one scientist from Israel. Okay, I think these 2 scientists or one of them came to Science Congress last year. Okay, they received a Nobel Prize for this, just recognize that name. So he figured out how ribosome actually reads the information of the mRNA. So now mRNA acts like a template in a way. So it's like reading bit by bit information on mRNA, adding amino acids corresponding to it. Okay, let me explain a bit on this and then we'll go to the video. Video will revise what I'm talking about and provide an image to you. So are you clear till mRNA? Because we already talked about DNA replication in detail. This should be easy for you to follow how mRNA is made, a complementary strand to the template 3' to 5' template. Next, we talked about protein synthesis. So mRNA came out looking for ribosomes. What does ribosome do? I think I'll show you an image for this. Yeah, this image is better. So translation, we're talking about translation here. So ribosome is a 2-part piece, 2 pieces that it has. Small unit we call, large subunit we call. So these two, between these two, mRNA is placed. Okay, these 2 pieces basically like this. mRNA is placed in the center and ribosome, top ribosome sits on top of it. Then what happens? So it reads not letter by letter. So far we talked about A, T, G, C. For A you add T, for T you add A. We talked about letter by letter. Here, ribosomes reads it differently because it needs to create variety. We only have 4 letters. We need to create more variety, right? So ribosomes reads word of 3 letters each time. So ribosome reads, as you see it here, it reads UUU, UCG, GUC. It reads a word of 3 letters. This is called codon. This is called codon. Sometimes we use the word triplet codon because it reads 3 letters at a time. That is what's called a triplet. Another Indian-born scientist discovered this. Hargovind Khurana. You ever heard about Hargovind Khurana? He was the one to tell us about this. That ribosome reads 3 letters each time. It's called triplet codon. Triplet codon. So ribosome is trained to read words made up of 3 letters. So ATG, GUC, CCG, like any 3-letter words. But as it is written on the mRNA, as you see here, in the mRNA, we talked about mRNA AUG, CGG. It reads AUG first. Then it reads CGG. Then it reads UAC on this mRNA. Then UUA. So it reads words like that. And for every such word, there should be a corresponding amino acid because proteins are made up of amino acids. AUG, we have an amino acid called methionine. For CGG, we have an amino acid called arginine. Like that, for this every word, there is a corresponding amino acid. It's like this, RAT, rat. Rat means you know what to look for. So rat is added to cat, RAT, CAT. You take a rat and then came the CAT in the transcript, you added a cat to it. You join rat to cat. Similar way, here they are joining amino acids. AUG corresponds to one amino acid, CGG corresponds to another, UAC corresponds to another. There are like that 20 standard amino acids. From bacteria to humans, these letters are fixed. It's 20 standard amino acids. So like 4 nucleotides, there are 20 such amino acids. There are extra amino acids, I think totally 23 or so, but 20 are common to all living forms. We call them 20 standard amino acids. Okay, you don't have to by heart those names. Just know that there exist amino acids. There are 20 standard of them, and there should be 20 such codes matching to this amino acid. Something more than that, I don't want to explain that complexity, but sometimes we have more codes to the same amino acid. Like let's say methionine, you may have AUG, AUC, AUT. So more codes for the same word. It's like in English, right? You may have multiple words for the same subject, right? So similarly, there may be more codes because if you mathematically work on this, because most of you are engineers, you can figure out how many codes can you create from 4 letters. A, T, G, C. If you want to create 3 letters, how many codes can you create? Using 4 letters in 3 different combinations. There's 3 combinations. How many letters you can create?
64.
64. How is it possible? How did you figure it out?
So by permutation combination, every place.
Permutation combinations. Correct? You can figure out from that there are 64 codes possible. There are 64 codes for it. If you read the material I posted on the orientation class on the cell DNA basics, they talked about the 64 codon story. If you're reading and if you get lost, this is what it is. Okay, so permutations and combinations that you can figure out. We are figuring out, we want to create 3-letter words from 4 existing alphabets. Correct? 4 power cube, right? 4 power 3, 4 into 4 into 4. 16 fours are 64. Correct? Okay, right. Plus, in addition to that, how does ribosome find amino acids? Let's say ribosome is read AUG. It read AUG like this here. This ribosome read AUG. But you know, it requires help of another RNA. I told you there are a variety of functions for RNA. The 2 remaining most of the important functions we see here, one is mRNA, messenger RNA. Second one we call is tRNA. You see this molecule called tRNA? tRNA refers to transfer RNA. mRNA refers to mRNA, messenger RNA. tRNA refers to transfer RNA. What does it transfer? It transfers. Do you see here? It is now bringing complementary code. This is mRNA in the picture. What is it doing here? This particular tRNA, if you notice, there is UCG. It has complementary sequence attached to tRNA. It is called AGC. Plus, it is bringing amino acid. Do you see it? tRNA is a transfer RNA for amino acids. Ribosome reads the letter and says, tRNA, can you please bring me amino acid corresponding to this? And tRNA picks up the amino acid again from our food. In our diet, we eat proteins of plants, we eat proteins of animals. They're degraded into individual amino acids. We don't use proteins of plant. We don't use hormones of animals. We create our own hormones in the body, because as soon as they enter in digestive system, they are degraded into individual amino acids. And those amino acids are raw material for creating our proteins. Who does them? Who brings them out? So ribosome reads the mRNA, asks for the amino acid, and tRNA is the molecule which grasps that amino acid, brings it to the mRNA, or brings it to the ribosome. Like that, tRNA molecules brings the amino acids corresponding to the code on the mRNA. And ribosome is the regulator. Ribosome is the catalyst. Ribosome is the enzyme. Third function for RNA. So far I told you mRNA, messenger RNA, tRNA, transfer RNA for amino acids. Ribosomal RNA. Ribosome is also mainly made up of RNA. This is RNA molecule mainly with proteins attached to it. So this is called ribosomal RNA. Ribosomal RNA is important for protein synthesis. So the three key functions of RNA, as opposed to one function of DNA, which is storing genetic material. RNA acts like a messenger, transfer RNA in synthesizing protein as a key enzyme. This is the only enzyme which is not made up of protein. It is made up of RNA. I don't know if you ever heard about RNA theory of creation of life. RNA world hypothesis. I don't know if you ever heard about it. Scientists thought RNA is more capable than DNA. Maybe at one point of time, RNA was a genetic material. Because of these features of RNA, they thought RNA is more diverse, more versatile. And I come from a lab. My PhD happened to be on this topic as well, because my supervisor was the one to figure out how this amino acid transferred. She comes from Max Planck Institute in Germany. They figured out how TNase actually receives these amino acids. They did a research on this, her supervisor. They figured it out, published in Cell, and I did PhD with her. We learned, my work is related to how tRNAs gets modified. My PhD was on this topic. Okay, right. So are you with me so far? We talked about how ribosomes synthesize it. Ribosomes reads the mRNA, 3 letters each time. This is called triplet codon. And every time it reads the 3-letter code, it brings the corresponding amino acids with the help of tRNA. You see tRNA coming here with the matching amino acid. And what is happening here? One side, it confirms, okay, the letters are matched now. I got corresponding amino acids to U, C, G in the form of A, G, C. So they match it. And now when match happens, amino acid will be added to the next tRNA. So what's happening? Amino acid, incoming tRNA bringing amino acid, right? So what happens? This amino acid is kept, tRNA is removed. So tRNA will leave the place, leave the ribosome, giving the amino acid to the first tRNA. Then what happens? Next tRNA comes with the amino acid. So second tRNA amino acid is added to it. Third amino acid is added to it. So chain is created. Just the way nucleotides are added by DNA polymerase, here chain is created by adding amino acids to each other.
So this tRNA has every kind of all the kind of combinations available and all kind of amino acid according to them.
Exactly. It has all combinations of those things, except out of those 64 you mentioned, for 3 tRNAs, we don't have codes. 61 can match. But imagine it cannot go forever, right? I told you we are only translating one small portion on the DNA called a gene. So meaning it has to end somewhere, right? You know what happens in the codes? You always see towards the end, there are some codes to which there are no amino acids, no corresponding tRNAs. There are 3 such things. If I remember it right, UAA, UGA, one more. I forgot exactly what was that. So there are 3 codons to which there is no corresponding tRNA, no corresponding amino acid. So what happens when it hits that tRNA, when it hits the mRNA nucleotide, where no tRNA comes in, reaction will stop. Translation will stop there. This is the end of the translation. Okay, so out of 64, there are for 61, we have corresponding tRNAs. Okay, so this is a process of protein synthesis, which happens in 2 steps: transcription, translation. Transcription happens with the help of, I didn't mention the enzyme here, but I mentioned before in the DNA replication. Can anyone guess how is the enzyme which makes mRNA called? I told you DNA polymerase creates DNA. What enzyme creates RNA?
RNA polymerase.
RNA polymerase. That's it. RNA polymerase. And ribosomes steal the second part, which is translation. Do you see how is it called translation? Because from DNA, information from DNA is being used to create proteins which are made up of amino acids. In a way, from the analogy, I can tell DNA is in German, protein is in English. Different alphabets. Amino acids here, nucleotides there. Because I remember one student said the DNA proteins are same. They're not. Someone said, I think, DNA is made up of proteins. They are not, right? DNA is made up of nucleotides. Proteins are made up of amino acids. And proteins are made based on the information we receive from our parents. You look like your parents because these define the phenotype. These define the characteristic. You have black color eyes because maybe your father or mother or both of them have black color eyes. Because the instructions you received will result in the protein which will give rise to black color eyes. Or, hairy, like a curly hair or straight hair. Your color.
Can you also say that all this is made from ribosomes? Because I think that is the key thing where everything is getting created.
But right? Ribosome is a translator. What it does is, whatever transcript you give it to it, it will read it, translate it. It is not defining the information. It is only translating the information.
But so where does the material come from, sir?
Where is the
material come from? Like every protein will be made of some structures, amino acids and everything.
Food, the diet. They say that, right? Doctors say if you don't eat proper proteinaceous diet, you won't be able to build muscles. Muscles are made up of proteins. Unless you give the raw material to the body, it cannot synthesize proteins. We're talking about in the last class, people who are malnourished. Do you think they build muscles? No, they cannot build muscles. Because the food provides the raw material, because we eat plant food, animal food carrying proteins, proteinaceous food, which is broken down into amino acids. Our ribosomes add amino acids as instructed by the mRNA. But the mRNA is designed or made from the DNA that came from our parents. So our creators in this sense are our parents. The instructions, good or bad, comes from them.
So would that mRNA transcription is foolproof, or here also can we expect some errors?
Usually the problems happen in the process of creating mRNA or in the DNA replication. Usually we don't find problems in synthesizing proteins. Proteins are more or less foolproof because they watch it and add it, and there is a variety of mechanisms to make sure that it is adding the right one. So errors usually introduced at the DNA level, not at the protein level. Okay, something else can go wrong. We'll talk about in genetic polymorphism. Genetic polymorphism, we'll talk about protein polymorphism. There I'll talk to you briefly what else can happen, how variety of proteins can be made from same RNA. It is not done erroneously. It is done because there are other methods to create proteins than just by reading like this. So it is like this. You want to read 3 letters, but what do you switch to the next letter? In the first letter itself, you made a mistake. You slipped. Instead of starting to read from AUG, you started to reading from UGC, GGU. What happens? Whole frame shifts. This is called frame shift mutation. We'll talk about that in polymorphism in a different class. Okay, right. So this is what is protein synthesis I wanted to learn. So we kind of figured out all the majority of the things you need for this class. Okay, just finish this with the video. 2 minutes video starts from I think last class we talked about histones, proteins which anchor the chromosome compaction. This 2-minute video starts from there to the end of what I discussed today. So very short and quick video. This is the power of animations.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called a nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
Could you follow that? I think one thing they mentioned in between that they have to process the RNA. That's a bit complex process. If needed, I'll talk to you about that. But for now, just imagine that it's a continuous process. So next thing they talked about, finally, once the protein is made, it has to undergo to form a proper structure. This is very, very important. We again study this in some diseases because of the improper structure of the protein, it doesn't function. So if there is a single letter that is being modified, it can result in a complete change in the shape. Not sure if you heard about sickle cell anemia problem. So problem happens here, only one letter has changed, one amino acid has changed, but it is sufficient enough to give rise to protein with abnormal structure. Abnormal structure results in absence of function, lack of function. So proteins with one letter change out of 560 letters there is enough to kill the function of the protein. One small change, which is called a mutation. Okay, we'll discuss more on the mutation, which is given in the syllabus. Mutation is the error that happens either in the DNA, but the mistake that gets created in the DNA. Either maybe because of the synthesis process or there are other methods for this. But change in the letter is called a mutation, it refers to mutation. Change in the DNA sequence is called a mutation. So if mutation happens in the key region of the gene, it affects the protein. If protein is affected, function of the body is affected. So we'll pick up many examples because we have dedicated chapter for this chromosomal aberrations. In many chapters, we'll talk about sickle cell anemia, hemophilia, Huntington's, many diseases you'll be introduced to. There we'll learn so much about mutations and their role in evolution. Okay, right. So this is what I want to talk to you today. With this, we finished talking about unit 1.7. Okay, so reading, I already told you there is one chapter in the orientation. I have not moved this to the book, to the course page. This is the PDF, so I don't have a Word document for that. But for the next class onwards, the outlines I give it to you where needed. I give them in the Word document so you can print and write on the side. But the material I'm going to give you is from the standard books, PDFs. So that information, I don't have a Word in the Word form, which you can print and read from it. Okay, I recommend that. Otherwise, I also gave you an additional resource for this unit. Class 12th NCERT book also talks about these concepts. So class 12th NCERT is very useful to majority of the topics in this class. Okay, so we'll pick up from class 12th, but it is more complex than what we need. So I'm just filtering it where needed. But otherwise, most of the information I'm providing in the classes comes from variety of sources, not just one or two, because I have knowledge in this field. So I pick up what is more easier for you to digest, what adds the value to your understanding. So I'm picking up like that. So majority of the information not taken from any single source. I don't use books like Pinath at all, because I prefer books should be better than Pinath, which is kind of a Bible for physical anthropology, I see, recommended by many specialists because they don't understand the subject very well, I feel. So we are going to pick up from various sources, and I'm going to give the actual sources to you and the summary I made from it, so that the experience you gain from these classes should be very rich. That is what I'm expecting. That is what I'm hoping. But get the maximum benefit from these classes through interaction.
That I have messaged you on Telegram the other day that I have taken classes on your stencil last year, and this is my repetition.
Oh, is it? Who is this? Who am I talking to?
Gayatri.
Oh, Gayatri. Yes. Okay.
Yes. So I think I am planning to listen again also. My only intention rejoining is that I want to write tests. As in your stencil course, there was no writing practice. It was only just one-way thing. I have listened all the classes and have made notes from the sheets you have given,
right,
and all. So what do you suggest? Should I listen classes or?
No, ma'am, I think I told you, I remember this. Unless if you're comfortable with the information, you don't have to listen or sit through my classes because it wastes a lot of your time, right? If there are any topics where you are uncomfortable, you want to listen to me again, because we evolve as the teachers every class and every session. So I may teach a little better in few classes here. So if you have any difficulty with few of the topics like that, do join the classes. Otherwise, you focus on writing. So I spoke to sir. I can actually give you the tests. So you can take the test I'm giving to the test series students, and you start writing answers to that. So wherever you're uncomfortable, follow my classes. Otherwise, you don't necessarily need to sit in my classes. Okay. Right, Gayatri. Did you find the classes useful, Gayatri? I think we have students with little bit of suspicion in mind sometimes. What do you suggest to students? How did you find the classes?
I find classes extremely helpful, sir, and I wonder why Feynman is not famous for anthropology. Why it is not recommended in
any
all
groups?
the anthropology circles. I have one day one of them were asking, what do you suggest? I posted positive comment, and then again I was worried. I just personally messaged that person, and it is good. I was telling them, but I don't see why Feynman is not talked.
Why is it not famous? Because equation is different. The way equation works in the market is very different. It requires a lot of promotion, a lot of advertisement, which we don't do. And especially our founder Venkat Mohan sir believes in bringing evolution in teaching. Okay, and we are not in the mode of attracting students. We don't want more students. We want great students who have that positive mindset, who wants to learn. So majority of the students, some students actually leave our classes too. That happened a couple of times where, because they said, sir, spoon feed us. Don't tell us all these things. Just tell us what to learn for the exam and go away. And we are not teachers who want those type of students. So we are happy with few students that we have. We definitely need to survive. The more we need more students on it. But the tricks that many institutions say, we don't try to play it. So we just want to just go with our teaching. If you like our teaching, promote them, come to a class. That's it. Okay. So
I find the classes extremely useful, and it simplified, sir. I could see previous questions and write answers to them, and I did not refer all, like you said, peanut. I did not see anything of that. I have seen your sheets and class notes only.
Yeah,
and
I spent enough time actually putting that information together. The information that we give in the class is very, very valuable. On top of it, there are definitely gaps you find. There are better ways of explanation you find. I must admit that. Then you can find other sources from that. For now, I just say that if you follow what we give in the class,
it is almost the preliminary, like starting, it is almost, I think 85%, 90% I found it can be done.
Okay.
There's just few random diagrams or something if we can collect that.
And there's nothing wrong in adding value from other books. I'm not against using them. But if I
no
believe,
time, no, sir, there is no time to refer that. Those very good.
Yeah, I believe, because I put my time into it, hard work into it, to make it to suit to the exam needs. Because I do analyze papers, look into how questions are asked, we analyze them, we conduct test series. So I go through all those phases of preparation towards the exam. So and my experience from this background helps me, because I am also science and technology faculty at various institutions like Braintree, La Excellence. I teach these areas there. So I look into the angle of science and technology, angle of physical anthropology, right? So we try to bring the best course possible from it. And we want to, I think we're working towards bringing the quality of teaching in this field. We want to raise that bar, okay? Which may take time to come into the limelight, but we are there. We're just playing the game. So soon, I think in the future, we'll be known. But we're not too worried if you're not commercially successful, because those students who come to our class, they say this exactly the same thing. They're completely satisfied.
The point is, before considering joining Feynman, you hear various names, and I just joined out of no choice, primarily to start with, because I had no choice on online. I had access only to this. So then either why people don't talk
Don't
about
talk about Feynman.
Okay.
I think you know the reason now, right?
Yes, right.
So spread it. If you really feel that someone can benefit from the class, I don't ask you to promote my name, but share this information with students. So we want actually more students to benefit, and even for us to commercially viable, we need more students in the classes.
So do you teach anthropology only here or any other?
Only here. I'm not associated with any organization yet. Some government organizations came forward to me, but I only teach one portion. So they always want one teacher to teach everything. And although I plan to teach the rest of the information from anthropology, which I'm just picking up, like archaeology and that, but still I teach with Venkat Mohan sir. Okay, slowly I think I share a little bit more from Venkat Mohan sir's burden because he wants me to teach more. But as such, not associated with any other organization in India. Not at the moment, at least. Okay, right, guys. I think I've already took enough time of yours, so I hope you are enjoying the classes. We'll stick to that. Ask me more difficult questions. We'll figure it out together, so I can even become a better teacher, which I want to. Right. So stay interactive, stay focused. Wherever you have a problem, I'll give the help on that. Okay, I'll bring more videos to you so that you can actually get this imagination better, which I didn't do much for my previous batches. But now I'm finding a little bit more time because I don't have to work on the basics anymore. So I can do more value addition to the classes. Okay, right, guys. Thank you very much. It is actually my pleasure having this discussion with you on these topics. Hope you got maximum benefit out of it. Okay, so we'll have alternate days, Wednesday and Friday. Next Wednesday and Friday, we'll have next classes. Okay.
Sure, sir.
See you everyone. Yeah. Bye.